Hello students, in today's class let's learn about the laboratory organization of an animal cell and tissue culture. The purpose of designing and development of an animal tissue culture laboratory is to stimulate natural conditions for growing cells outside of its natural environment, because very often cells do not behave or proliferate in vitro in a way that they would normally do in vivo. So in today's class, we're going to learn about planning a laboratory facility, design and layout, and disaster management. Planning. If we are constructing a new building, then compliance with the planning department will be required. Also, compliance with local and national safety regulations will also be required. While planning an animal tissue culture lab, main emphasis is on maintaining asepsis, that is, maintaining a sterile environment which is free of any biological or particulate contaminants. So rooms should be designed for easy cleaning, and distribution of equipment should be arranged in such a way that the equipments that create draft or dust should be located as far as possible from the sterile working area. Now, tissue culture lab should be separated from the preparation, wash-up, and sterilization areas while still remaining adjacent, and this is to avoid contamination as much as possible. The basic requirements for an animal cell culture laboratory must include sterile handling area, carts for servicing sterile handling area, pump and reservoir for withdrawing and discarding media, containment and quarantine areas, waste disposal for potentially biohazardous materials, regular waste disposal for plastic wrappings, used paper towels, swabs, tissues, etc., sinks for washing hands, incubation, provision for carbon dioxide, supply to incubators and gassing chambers, cell counting, centrifugation, wash-up for glasswares and apparatuses, water purification units, preparation and bottling of liquids, packaging plastics, sterilization facility by dry heat, steam and filtration, sterile storage, inverted microscopes for monitoring cultures, and high-power microscopes with fluorescence. The requirements depend on the scale and size of the laboratory and the number of users. The basic services for an animal cell culture laboratory would include provision for hot and cold water, electricity and power backup, carbon dioxide, compressed air, combustible gas, and also adequate flow drainage should be provided in the preparation wash-up area, vacuum line and pump. A vacuum line can be useful for evacuating culture flasks. Ventilation. Ideally, a tissue culture laboratory should be at positive pressure relative to the surrounding work areas, and this is to avoid any influx of contaminated air from the outside. If containment is required, it must be at negative pressure relative to the surrounding areas. To satisfy both requirements, it is preferable to have a positive pressure buffer zone receiving HEPA filtered air outside the tissue culture laboratory, such as the preparation area and microscope room or the corridor. Here we have a basic layout of a small tissue culture lab and tissue culture lab with adjacent preparation room. Here the dark colored circles indicate HEPA filtered air and the lighter colored circles indicate standard filtered air. Laminar flow hoods should be vented to the outside to provide most of the air extraction required for the room and to ensure that the incoming air, either from a central plant or an air conditioner, does not interfere with the integrity of the airflow in the cabinet. While setting up an animal cell culture lab, we need to ask certain questions, like how many would be working in the lab and for how long? What is the scale of the work to be done? How much space would be required for each facility? like for culture area, preparation area, wash-up, storage, etc. Answers to these questions determine the design and layout of the laboratory. Now, there are 6 main functions that need to be accommodated: sterile handling, incubation, preparation, wash-up, sterilization, and storage. Now, if a single room is used, we need to create a sterility gradient. The clean area for sterile handling should be located at one end of the room, which is farthest from the door, and wash-up and sterilization facilities should be placed at the other end of the room, with preparation, storage, and incubation in between. Sterile handling area should be located in a quiet part of the tissue culture laboratory and should be restricted to just tissue culture. That is, it shouldn't be shared with any chemical work or work on microorganisms like bacteria, yeast, or protozoa. The sterile working area is provided by the laminar flow hoods and biosafety cabinets. Laminar flow hoods as well as biosafety cabinets provide a working area with greater control on maintaining sterility as compared to a sterile room. Biosafety cabinets might be confused with a laminar hood, as both of these pieces of equipment work as enclosed workspaces with a ventilated hood and are provided with HEPA filters that decontaminate the air moving out of the cabinet. But laminar hood only provides protection to the sample and not to the personnel and the environment. On the other hand, Class 2 biosafety cabinet provides protection both to the sample and to the personnel, and Class 3 biosafety cabinets provide protection to all three: the sample, the personnel, and the environment. Most tissue culture laboratories will probably opt for Class 2 biosafety cabinets, especially when dealing with human cell lines. The service bench, ideally fitted with drawers and open shelves above, should be provided for the storage of sterile glassware, plastics, pipettes, screw caps, syringes, etc. The bench is also convenient for placing a microscope, cell counter, small centrifuge, etc. near the sterile handling area. Now, quarantine and containment areas. Newly imported cell lines or biopsies can be handled here until they are shown to be free of contamination, particularly mycoplasma. Incubators and warm houses. A carbon dioxide incubator with a humid atmosphere might be required for incubation of cell cultures. Generally, large numbers of flasks or large volume flasks that are sealed are best incubated in a warm room, whereas open plates and dishes will require a humid carbon dioxide incubator. Preparation area. In small labs, extensive media preparation can be avoided if there is a reliable source of commercial culture media. Although a large laboratory may still find it economical to prepare its own media, it is much more convenient to purchase ready-made culture media for smaller labs. All the items required for media preparation are to be sterilized. Then also, space is required for wash-up. The need for an extensive wash-up is minimized if disposable plastics are used, but there will still be a need for some glassware washing, as well as handling and disposal of used plastics, which may need to be autoclaved. Wash-up and sterilization facilities should be placed outside of the culture area to prevent humidity and heat buildup. Most preferably, autoclaves, ovens, distillation units should be placed in a separate room. Ample bench space is also required for handling of baskets of glassware, sorting pipettes for sealing, and packaging for sterilization. Wash-up sinks of proper size should be fitted with 4 taps for cold water, cold hot water, for deionized water, and one connected to a rinsing device. Now, storage. Storage facilities must be provided for sterile liquids and reagents, sterile and non-sterile glasswares, sterile disposable plastics, apparatuses, disposable items, liquid nitrogen, and carbon dioxide cylinders, etc. Sterile and non-sterile items should be kept separately and clearly labeled. And storage must be provided at room temperature for plastics, glassware, gloves, water, salt solutions, at 4 degrees Celsius for working strength and concentrated media, at 20 degrees Celsius for serum, glutamine, antibiotics, etc., and at minus 196 degrees Celsius for cryo-freezer storage. This diagram is a suggested layout for a simple self-contained tissue culture laboratory which can be used by 2 or 3 persons. The dark shaded areas represent removable equipment and the lighter shaded areas represent fixed or movable furniture. Now, since a single room serves as a laboratory, a sterility gradient is being created as mentioned earlier. Here we see that the washup area is placed next to the main exit and the sterile handling area is placed in the other end of the room. The preparation and storage area lie in the center, thus creating a buffer zone. This layout is for a medium-sized tissue culture laboratory. Now, this is suitable for about 5 to 6 people working. The washer preparation facility is located elsewhere here. Now, the sterile handling area is slightly larger so that it can accommodate more people. We have here 3 Class 2 biological safety cabinets. Now, lastly, let's study one very important aspect of an animal cell culture facility: disaster management. Ideally, an animal cell culture laboratory should have a central alarm panel, and it should be visible from the entryway into the building or in any other communal areas. The procedures to carry out and emergency contact numbers should be posted beside these alarm panels. Major catastrophes such as power outages, major fire, earthquakes, and floods should also be considered. Irreplaceable stock should be duplicated off-site in another laboratory or storage facility. Cell lines can be lodged with a reputable cell bank, for example, in case of crisis like widespread microbial contamination or cross-contamination. And valuable cell line data should be backed up locally on a routine basis and also off-site, either in another laboratory or in cloud storage. Now that's all we'll be covering today. See you in the next class. Thank you.